Hello and welcome back again to our discussion of, hold on a minute, hold on, I've got to actually start screen sharing here. Okay, there you go. Hello and welcome back to our discussion of chapter 27 on bacteria and archaea. And if you remember in part one, we ended with a discussion of conjugation and the transfer of an F plasmid. This basically this whole process that's summarized at the top of slide 46. Now we're going to look at conjugation and the transfer of part of an HFR bacterial chromosome that causes recombination. And so we'll jump into the discussion and the lecture right here at slide 50. So basically then what's happening is that a cell with the F factor built into its chromosome functions as a donor during conjugation. And such cells are called HFR cells for high frequency of recombination cells. The recipient cell becomes a recombinant bacterium because it has basically DNA from two different cells. So the first step then is the HFR cell, donor cell that has the F factor in its chromosome. Basically it establishes a pilus between it and an F minus cell. And then a single strand of the F factor breaks, detaches, is replicated from the original the original chromosome, and it begins to move through the bridge to the recipient cell, and then crossing over can result in exchange of homologous genes. And so, the A plus gene may be exchanged for the A minus gene in the recipient chromosome. And as a result, then the recipient chromosome now has the A plus gene. And then enzymes degrade and break down the DNA that's not incorporated into the recipient cell. And the recipient cell now is a recombinant cell. It's still F minus, but it's a recombinant cell because it has this new A plus gene, plus maybe a couple other things as well. And so that's conjugation then. Now let's look at R plasmids and antibiotic resistance. So R plasmids then are plasmids that carry genes for antibiotic resistance. So the R is for resistance then. So antibiotics kill sensitive bacteria, but not bacteria with specific R plasmids in them. So then through a process of natural selection, the fraction of the bacteria population with genes for resistance increases in a population that's exposed to antibiotics. So a lot of our plasmids then have genes that have the codes for producing pili, making it possible for resistance genes to be transferred between bacterial cells. Some R plasmids carry genes for resistance to multiple antibiotics. And so antibiotic resistant strains of bacteria are becoming more common, unfortunately, and the infections they cause are now harder to treat. So we have to continually find new antibiotics because bacteria are becoming resistant to existing antibiotics. So now we can look at diverse nutritional and metabolic adaptations. So diverse, in other words, a wide variety of nutritional and metabolic adaptations that take place in prokaryotes. So prokaryotes can be categorized by the way they obtain energy and the way they obtain carbon. So for example, phototrophs. Remember, troph means feeding, and phototrophs, that is light. So they obtain energy from light. Chemotrophs, chemo means chemicals, so chemotrophs get their energy from chemicals. Autotrophs basically can feed themselves. They do require carbon dioxide and some related compounds as a carbon source, but other than that, they can feed themselves, and usually they're phototrophs. Heterotrophs then need, hetero means other, and so they need an energy source other than just themselves. So they require organic nutrient to make organic compounds and to get energy. So energy and carbon sources are combined to give four major modes of nutrition: then photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. And you can see, so basically then the autotrophs, photoautotroph then has light as its energy source, and carbon dioxide and bicarbonate and other related compounds. And so photosynthetic prokaryotes are an example of these. For example, the cyanobacteria that we'll look at a little later, and also plants and certain protists like algae. And we'll look at those. We'll look at protists in the next chapter. So something to look forward to. And the chemoautotrophs then, basically this type of nutrition is unique to certain prokaryotes, and we'll talk about those a little later. The photoheterotrophs then are basically certain aquatic salt-loving prokaryotes. And chemoheterotrophs then, there are a lot of prokaryotes that are chemoheterotrophs. For example, and some protists, and then the fungi, animals, and some plants are chemoheterotrophs. And there's the same table in a little more detail. So then oxygen, the role of oxygen in metabolism, and the role of oxygen can actually be used to actually categorize a lot of living things into different categories. So there's a lot of variation in prokaryotic metabolism. So there are obligate anaerobes, obligate aerobes and obligate anaerobes, and facultative anaerobes. Obligate means they're required to do something, and aerobe means with oxygen. So obligate aerobes mean they require oxygen for cellular respiration. They can't perform cellular respiration without oxygen. It's required. But obligate anaerobes, so anaerobe means without oxygen. So they're obligated to, they're required actually by their metabolism to live in areas that do not have oxygen, because oxygen is actually a poison for them. So they live by fermentation or other things that will give them energy, and they perform anaerobic respiration then. Break down food to get energy without having to use oxygen for those processes. And facultative actually means it's optional. So facultative anaerobes means they can either live with or without the presence of oxygen. So they can use oxygen if it's present, or they can carry out fermentation or other anaerobic respiration then if oxygen is not present. Three major categories then. And then nitrogen metabolism is important as well, because nitrogen is essential for production of amino acids, because amino acids have nitrogen in their molecular structure, and it's also essential for nucleic acids as well, the RNA and the DNA, because they the nitrogenous bases, have a good amount of nitrogen in their molecular structure. So prokaryotes then. So prokaryotes can metabolize the nitrogen in a variety of different ways. For example, some prokaryotes convert atmospheric nitrogen, which is basically exists as N2, two nitrogens attached to each other. They convert atmospheric nitrogen out of the air to ammonia in liquid form, and that is called nitrogen fixation. It's called fixation because it's put into the nitrogen is put into or fixed into an organic compound that is useful and that can be used by living things, especially plants. Then there's also a lot of symbiosis, a lot of cooperation. The cooperation between prokaryotes, different types of prokaryotes, allows them to use environmental resources that they could not otherwise use as individual cells. Like the cyanobacterium, and cyano actually means blue. So this, like the chemical cyanide is actually a blue chemical. So the cyanobacteria, the blue bacteria, and actually they're the blue bacteria or blue-green bacteria, one called Anabaena. It's got photosynthetic cells, and it's also got nitrogen-fixing cells that are called heterocysts, or maybe heterocytes. And they exchange metabolic products. So in other words, the photosynthetic cells can actually produce energy by catching light from the sun, and they can supply the nitrogen fixing cells with energy, with ATP produced by photosynthesis, and then the nitrogen fixing cells can supply the photosynthetic cells with nitrogen, and so it's a very nice exchange, a very good symbiosis then. And here you can see most of the cells in Anabaena are photosynthetic cells, but there are a few heterocysts here and there that are actually nitrogen fixing. So Anabaena is a really so this is an example of prokaryotes forming colonies, and the Anabaena colonies are examples, very good examples of very successful prokaryotes, and they're often very common in sloughs and ponds and things. Freshwater. So metabolic cooperation occurs between different prokaryote species in colonies that coat the surfaces of things. And these surface coating colonies are called biofilms. And biofilms can be on the surface of a solid object like a rock or like bark on a tree, or these biofilms can also can form on the surface of aquatic habitats, especially on the surface of ponds and sloughs, and sometimes lakes as well. So sulfate-consuming bacteria and methane-consuming bacteria on the ocean floor can use each other's waste products as food. So sulfate-consuming bacteria produce methane as waste, and they're used by the methane-consuming bacteria. And the methane-consuming bacteria produce sulfate compounds as waste, and they can be used by the sulfate-consuming bacteria. So a pretty good trade-off then. So prokaryotes then have actually diversified into a large variety of different life forms. The prokaryotes now inhabit every environment known to support life, like we've mentioned, and advances in studies in genomics, studies of genomes, are beginning to reveal that, show that the extent of the wide extent of prokaryote diversity. And so, let's look at an overview. Genetic analysis then has led to our dividing of prokaryotes into two domains, the bacteria and the archaea. And advances in molecular techniques. Now, when they say molecular techniques, or when they say molecular biology, what they're meaning is genetic analysis, some sort of analysis, some sort of techniques to manipulate and analyze DNA and RNA. So advances in molecular techniques then help to add new branches to the phylogenetic tree of life for bacteria and archaea, these prokaryotes, each year. And so the use of a good DNA technique called polymerase chain reaction, or PCR, has allowed for rapid sequencing of prokaryote genomes. And a handful of soil may contain 10,000 different prokaryotic species. And horizontal gene transfer, that's transfer from one species to another between prokaryotes, can obscure the root of the tree of life. So they do a lot of transfer and a lot of mixing and matching of genes and things. So the bacteria then, as a domain, include the vast majority of prokaryotic species that are familiar to most people. Then, and all of these prokaryotic species in the bacterial domain exhibit a diverse nutritional type. Diverse nutritional types. So, and it seems like, according to people who look into the phylogeny of and evolution of things, it looks they're thinking, according to evolutionary thoughts, they're thinking that maybe the bacteria look like they branched off of the others before the eukarya and archaea branched off. So there's an alpha subgroup, a beta subgroup amongst the bacteria, also a gamma subgroup, a delta subgroup, and an epsilon subgroup as well amongst the bacteria. And so let's take a look at a few of these then. So the proteobacteria then, are gram-negative bacteria that have photoautotrophs, chemoautotrophs, and heterotrophs. Some are anaerobic and others are aerobic. There are currently 5 subgroups of proteobacteria that are recognized. So there's the subgroup alpha proteobacteria, and that's basically all these pictures that we looked at here. Alpha proteobacteria, a lot of species are closely associated with eukaryotic hosts, so they're parasitic. And maybe, those that hypothesize about, evolution and origins of things think that maybe mitochondria actually evolved from aerobic alpha proteobacteria and through endosymbiosis. So endosymbiosis basically is a symbiont that is actually inside another species. So an example is rhizobium, then. It forms root nodules in legumes, and it fixes atmospheric bacteria and atmospheric nitrogen. Legumes are basically the group of plants that includes all pea plants and all beans then. So peas and beans have root nodules with rhizobium in them that are able to fix atmospheric nitrogen. So a legume plant never has to worry about a shortage of nitrogen because they actually have rhizobium bacteria inside them that will just pull nitrogen right out of the air. So a legume never has to worry about nitrogen. Then agrobacterium. Agrobacterium then produces tumors in plants, and it's used in genetic engineering of plants. And so here's a good example of rhizobium. So the arrows show the presence of rhizobium bacteria inside a root cell in a legume. And TEM means this photograph was taken at a very high magnification by a transmission electron microscope. So TEM is transmission electron microscope. In the beta subgroup of proteobacteria, it's nutritionally diverse. Then, and one example is a bacteria that are in the soil, Nitrosomonas. And Nitrosomonas then converts ammonia actually to nitrous oxide. And nitrous oxide then can be used by a lot of different types of plants. Other members of this group include aquatic species and also pathogens, some that cause disease. And so here's Nitrosomonas then. Again, transfer electron microscope photograph. So it's a little over 1 micrometer wide, so very, very small. Then there's the gamma group, the gamma proteobacteria group. They've got autotrophic members that include sulfur bacteria, such as Thiomargarita. How about that for a name? Thiomargarita namibiensis. So it's called namibiensis probably because it comes from Namibia, or it was first found in Namibia. And so, but there are also some heterotrophs in this gamma group that are pathogenic. So Legionella that causes Legionnaire's disease, Salmonella that causes the disease called Salmonella, and Vibrio cholerae that causes cholera. And then also in this gamma group then are Escherichia coli or E. coli that's in the intestines. It's a normal and natural resident inside the intestines, usually large intestines, of many mammals. And it's not normally pathogenic if it's in the intestine, usually large intestine. So here's Thiomargarita namibiensis that has sulfur waste in it. And so LM, this is a light microscope. So just a compound light microscope then. And these are a lot bigger than the Nitrosomonas then. And so here's the delta subgroup of the proteobacteria. A good example are the slime-secreting myxobacteria. And the myxobacteria produce a drought-resistant myxospore. And then another example is the Delavibrios. The beginning B is actually silent. So the Delavibrios that basically do high-speed attacks on other bacteria. And then, so here's some Chondromyces coractus type of myxobacterium then. And this is done by scanning electron microscope, the scanning electron microscopes. Then epsilon proteobacteria. Most of the species in the subgroup are pathogenic. And so Campylobacter then, that causes blood poisoning. And also Heliobacter pylori, which causes stomach ulcers. Heliobacter pylori. And here's a nice color photograph of Heliobacter pylori then. And then there are the chlamydias. So they're parasites that live inside animal cells. And a good example is Chlamydia trachomatis. And this Chlamydia trachomatis then causes blindness and causes non-gonococcal urethritis transmitted sexually. And here are some chlamydia, then the arrows show chlamydia inside animal cells. Then there are spirochetes. So they're spiral shaped, they're helical shaped gram-negative heterotrophs. Then some are parasites, like the Treponema pallidum that causes syphilis, and others the Borrelia burgdorferi then, which causes Lyme disease. That's transmitted by ticks, remember. So here's a nice, beautiful color photograph taken under a transmission electron microscope at very high power, showing a very nice, couple of very nice spirochetes then. And then now these cyanobacteria. Remember, cyano means blue, so they're gram-negative photoautotrophs. And those that hypothesize about evolution are thinking maybe that plant chloroplasts actually evolved from cyanobacteria that began living inside cells as endosymbionts. Remember, endo means inside, and symbiosis or symbiont means a species that lives in close association with another species, not always inside the other species. So that usually not, most cases of symbiosis is not where one species is living inside the other. So they're in large numbers then, and they're large components of freshwater and marine communities of phytoplankton. Plankton, basically, if you remember plankton from SpongeBob, in SpongeBob, plankton is very small. The little character plankton is very small. And so plankton in biology are very small organisms, usually microscopic or close to microscopic, that are in the water. And phytoplankton are plankton that are plant-like and perform photosynthesis. And here are some cyanobacteria called oscillatoria, and they're filamentous. They produce colonies that are very long, thin filaments that are just a long, thin chain of many, many cyanobacteria cells connected together in a big chain of a colony. And you've got a video of oscillatoria, and you can watch that video on your own time. You're kind of turning there and twisting a number of different things. Kind of an interesting thing. And so there are a number of gram-positive bacteria as well. But I think let's look quickly at the gram-positive bacteria. Gram-positive bacteria, this group includes colony-forming groups, like things called actinomycetes, many of which help decompose organic matter. There are also solitary species that are not colony-forming, like Bacillus anthracis, that causes anthrax, or Clostridium botulinum, that is the major cause of botulism or food poisoning. There are different types of Staphylococcus that cause staph infections, and Streptococcus that cause strep infections, like strep throat. There are mycoplasmas. Myco means fungus, and they're the smallest known cells, and they have no cell walls. And so here's some Streptomyces bacteria, kind of interesting shapes. And here's some mycoplasmas, hundreds of mycoplasmas, very, very small mycoplasmas, covering a human fibroblast cell. The fibroblast cells are in connective tissue in the human body. And so that's basically a quick summary then, up to page or up to slide 95. That is basically a quick summary of domain archaea, or domain bacteria. In the next lecture, that will be part 3 of the next lecture. That is going to cover the domain archaea. Start with a good coverage of the domain archaea. So then remember, always check your emails and always check your Canvas for announcements and things to keep up with whatever's happening in this class and other classes. And so I will say goodbye for now, and I'll see you during part 3, the last part 3 of the discussion of chapter 27 in the biology book textbook. Thank you and good night.